Okay, so welcome to this next video in which we are discussing angiogenesis in chronic inflammation. Okay, so we're currently looking at the effect of the angiogenic factors on an endothelial cell. Okay, so we've seen that the two major angiogenic factors, vascular endothelial growth factor A and fibroblast growth factor 2, are going to activate receptor tyrosine kinases. Vascular endothelial growth factor A will activate the vascular endothelial growth factor receptor 2, and fibroblast growth factor 2 will activate the fibroblast growth factor receptor 1. Okay, and these receptors then undergo activation, and this results in this cross-phosphorylation, which results in both of the members of the dimer having phosphotyrosine residues on their intracellular domains. These phosphotyrosine residues are then going to lead to many downstream pathways, three specifically. Three that we're going to look at anyway. One was the RAS pathway, which led to the activation of the MAP kinase enzymes. Okay, the second we're looking at now is where you activate this phospholipase C gamma enzyme, which is usually within the cytoplasm of the cell, but when it has these phosphotyrosine residues here on the intracellular aspect of these receptor tyrosine kinases, it's going to come and associate here, and when it binds here, that's going to mean that it's now very close to the membrane, basically. Whoops, sorry, you can't see this. Okay, so when it comes and binds here, it's going to end up very close to the cell membrane, and it can then start breaking down phosphatidylinosytol 4,5-bisphosphate into diacylglyceride and IP3. Okay, so what does IP3 now do? Because it's been released into the cytoplasm of the cell. Well, basically, it's going to come and act on receptors which are in the cell, which are in the membrane of an intracellular organelle, which specifically is the endoplasmic reticulum. Okay, so within cells, you have an organelle known as the endoplasmic reticulum, which is called the ER for short. So this is the endoplasmic reticulum. And the endoplasmic reticulum is a membrane-bound organelle, okay? And inside the endoplasmic reticulum, you have a very large store of calcium, okay? So inside here, you have a lot of calcium. Okay, and I might even just bring this round like so to make it look more convincing. Okay, and in the cytoplasm of the cell, you have very low levels of calcium, around 100 nanomolar calcium. Okay, so the cytoplasmic calcium is 100 nanomolar concentration. Okay, and if you activate these IP3 receptors, as they are called here, by binding IP3 to them, what this will lead to is the opening of these IP3 receptors. Now, the IP3 itself does not actually open them, but it gets them ready to open. And if you're interested in the details, I have an entire playlist on calcium signaling in which we go into this in huge detail. Okay, so IP3 is going to bind to these IP3 receptors. Specifically, 4 molecules of IP3 will bind to the IP3 receptor, and it will lead to the opening of the channel. It won't open it itself, but it will lead to the opening of the channel. And then what will happen is you'll get release of calcium from the lumen of the endoplasmic reticulum into the cytoplasm of the cell. Okay, so now what does this calcium cause? Well, basically, it's going to bind to another protein and activate this protein, and then that protein will bind to another protein and activate that protein. So, calcium is going to bind to a protein known as Calmodulin, okay, which is often abbreviated as CAM. So let me just show you the structure of Calmodulin. So Calmodulin has these two lobes, okay? It has a lobe known as the N lobe and a lobe known as the C lobe. Okay, so this is the N lobe here, and this is the C lobe over here. And let me just bring this up a little bit. C lobe. Okay, and on each of these lobes you have 2 calcium binding sites. So the N lobe has 2 calcium binding sites, and the C lobe has 2 calcium binding sites. And this structure at the moment, where you have calmodulin with no calcium bound to it, is what's known as apocalmodulin. Okay, and the shorthand for calmodulin is to write this capital C, lowercase a, and then capital M. So apocam is the shorthand for apocalmodulin. Okay, so you can see that you have this sort of hunched over structure where the 2 lobes are sort of hunched over close to one another. Now when calcium binds, i.e. when 4 calciums bind to the calmodulin protein, 1 in each of these calcium binding sites, what it causes is it causes the 2 lobes to move out from one another. Okay, so these 2 kind of sort of flip out away from one another. And then the linker, the polypeptide linker between them, goes from being a linear polypeptide of amino acids to having an alpha helical structure. Okay, so this is an alpha helix here. Okay, and this is the change which occurs when calcium binds to these 4 calcium binding sites here. So let's colour in the calcium in turquoise. So these turquoise dots represent the calcium bound to the calmodulin. And this structure now is what's known as a calcium-calmodulin complex. And this can go on to activate a whole bunch of other proteins. Okay, and it's often abbreviated shorthand to Ca2+ for calcium, and then CAM for calmodulin. Okay, now calcium-calmodulin complexes activate an enzyme known as calcineurin. Okay, so let's put calcineurin here. So this is our enzyme calcineurin, which, by the way, is often abbreviated as C, and then you put again lowercase a and then N afterwards rather than M. So this is calcineurin. Okay, right. And then bound to the calcineurin is now this calcium-calmodulin complex here. Okay, right, and there will be two calcium ions bound to each of the calcium binding sites on each of the lobes of calmodulin. So 4 overall. So we'll colour in calcineurin in pink here. Okay, so now what does calcineurin do? Well, basically, calcineurin is what's known as a serine-threonine phosphatase. It removes phosphate groups from serine and threonine residues. So we have talked a lot about serine-threonine kinases. We saw that horrendous pathway which had just serine-threonine kinase after serine-threonine kinase. Now we've got a serine-threonine phosphatase which is going to remove phosphate groups off serine and threonine residues. Now you might think, well, isn't that just going to reverse all of the things that the other ones were doing? And isn't this just totally the converse of what we had before? Well, you have to remember that this has certain targets. It doesn't just remove serine and phosphate groups from serine and threonine residues on anything. You have to be a certain protein for it to work. Now, which protein is it going to target? Well, it's going to target a protein known as NFAT. And NFAT is another transcription factor, okay? And this time the removal of phosphate groups from this transcription factor is going to activate it, whereas we saw with ELK1 and MYC that it was the addition of a phosphate group that activated it. So this is the transcription factor NFAT, okay? Which stands for the nuclear factor of activated T cells. So the nuclear factor of activated T cells. And basically, this nuclear factor of activated T cells has a region known as the serine-rich region, which contains a whole bunch of serines, which are usually phosphorylated. Okay, so let's say that this here represents the serine-rich region, which by the way is often abbreviated to SRR for short. So serine-rich region, SRR for short. And this has a whole bunch of serine residues in, which are usually phosphorylated, okay? So all of these serine residues will be phosphorylated. And basically the phosphorylation of these serine residues is keeping this N-fat protein in the cytoplasm of the cell and stopping it from going into the nucleus. Okay, so if you remove the phosphate groups from this serine-rich region, then the N-fat can suddenly move from the cytoplasm into the nucleus of the cell. And then once it's in the nucleus of the cell, it can act as a transcription factor, i.e. it can bind to the promoter regions upstream of genes and increase the transcription of those genes. Okay, right, so this is what calcineurin is going to do. Calcineurin, or CAN for short, is going to come and remove the phosphate groups from those serine residues. And then the nuclear factor of activated T cells will be able to move from the cytoplasm of the cell into the nucleus of the cell and act as a transcription factor. Now, what is it going to increase the expression of? Well, the answer to that is it's going to increase the transcription of molecules or proteins that are involved in taking the cell into the cell cycle, basically. I.e. it's going to do the same thing as MYC and CFOS-CGen heterodimers. So we've got another one of these transcription factors, which we're going to discuss at the end. I.e. we're going to reconverge all of the pathways at the end, and here's another one that we will be able to add in. So this pathway again has led to the activation of a transcription factor, which is going to take you from interphase into the G1 phase of the cell cycle. But I'll go through that at the end. Okay, so there is one final pathway that we need to look at now, which can also be activated by these receptor tyrosine kinases, and which is going to lead to the activation of growth or cell division within these endothelial cells. Okay, and we'll look at that pathway in the next video.